Good morning children, continuation of DNA fingerprinting. Now we will learn how to separate the normal DNA from that of the sad DNA. This will be done with the density gradient centrifugation. When this is done, the bulk DNA will be the major peak and the smaller pieces will be of the sad DNA, which will consist of the repetitive DNA sequences, which forms the large portion of the human genome. And second point is that they show polymorphism. What is polymorphism? Different morphology, which will be because of mutation. And we know as they don't affect they don't code for any of the major proteins, so it will be passed to the next generation. And when it is found in the large number of generation, then it becomes polymorphism. That means if the frequency is more than 0.01, it becomes polymorph. It is said to be polymorphism. Now, this is the basis of the DNA fingerprinting. One thing you should understand that polymorphism will be same for all the tissues of an individual, like saliva, hair, bone, whatever. Now take an example. Suppose this is the murderer's DNA and this is the suspect 1 and suspect 2. So all the chromosomes are studied, but for example, now here I am explaining for chromosome number 1. So homologous pair of chromosome, murderer, this one is having 5 repetitive sequences, this one 2. This suspect 1 is having 8 and 6. Suspect 2 is having 5 and 2. So you can see it is matched. But it's not so easy. It's a complex process. The methodology will be that first the isolation of the DNA has to be done. This chromosome is present in the nucleus. So different types of enzymes like lipase, proteases, amylase, RNAs will be used to digest them. Then chilled ethanol will be added and DNA will be separated. Next is that whole of the DNA we don't need. So we have to cut the repetitive pieces of DNA, which will be done with the help of restriction endonuclease enzyme. Now this will be added to the gel electrophoresis process. The gel is composed of agarose gel, and this part is anode and this is cathode. We know that DNA is negatively charged, so the pieces will be attracted towards the anode. The smallest pieces will be the farthest. Still we cannot see this DNA, so the next step will be that nitrocellulose paper will be placed here, and this process is known as Southern blotting. After that, radioactive VNTR probe will be added to it. What is this? This is complementary to the sequence. Like if the sequence is 80, 80, 80, then the VNTR probe will be TA. So a hybrid will be formed. Rest of it will be washed away, and then last step will be autoradiography. And this will be done with the help of X-ray. And now it will be clearly visible and it will be easy for the detection of the murderer. Now suppose on the murder scene, if we get a very small amount of piece, then how to complete the experiment? Before doing all these methodology, PCR is done, polymerase chain reaction is done, so that that portion becomes in a large number, and then this again methodology will be followed and the suspect will be clear that who has done the murder.